disease in cauliflower is used. This viral gene is also known as the promoter gene, namely CAMV35S, forces the host organism to take up the inserted gene and forces the host to produce the GE protein at a very high level in every part of the host plant or animals. Another piece of genetic fragment, known as the terminator, is also attached to this construct and signals the end of the genetic constructs. From here on, the science becomes very imprecise. This construct, otherwise known as the gene cassette, is forced into the host cell by means of various methods. In one method, an instrument called the gene gun is used. In this method, gold or tungsten particles coated with the gene is blasted into the host cell. Other methods like electroporation, sonication, and Agrobacterium tumefaciens are used to invade the host cells with the desired genes. These methods are totally unpredictable and imprecise. As it is not possible to know which cells took up the desired genes, another gene known as the ARM gene or the antibiotic resistant marker gene is also attached to this construct. When antibiotic is applied to the cellular aggregate, only those cells which took up the genes survive, thereby marking the engineered or the transgenic gene. These modified cells are grown in tissue culture mediums into plant with the desired characteristics. The use of bacterial, viral, and antibiotic-resistant genes in the modification of these organisms opens up the possibility of creating dangerous organisms which are a threat to human health, the ecology, and the environment. The health of humankind is at greater risk than ever. For example, the promoter gene has the capacity to alter the constitution of the host by switching off good genes and switching on harmful genes and thereby promoting the production of anti-nutrients, allergens, and toxins. The antibiotic-resistant genes, if transferred to other disease-causing bacteria, confer antibiotic resistance on them.
This is one way bacteria adapt. A bacterium with a plasmid, plasmids hold genetic information, will connect with another bacterium. A copy of the DNA is transferred. The new DNA may dictate a new food or energy source or tell how to dodge an antibiotic. This sharing is passed on and on to other bacteria, even among different species, and it can happen in less than an hour.
I think contributes to the problem of resistant bacteria in the community, which then becomes a problem in the hospital, which then becomes a problem in extended care facilities, and they're all inextricably linked.
This method of artificial horizontal gene transfer also opens up the possibility of creating new disease-causing viruses and bacteria. In fact, new diseases are being created every day, which weren't there for millennia. One such disease that is afflicting tens of thousands of Americans is the Morgellons disease. It is a very painful disease, similar to Lyme's disease and scabies. It is so painful, and the sensation of live things crawling under the skin has forced many to commit suicide in desperation. Morgellons has no known cure. Agrobacterium tumefaciens, the bacterium that is commonly used in genetic engineering, is also the cause of this disease.
I have one theory. The type of bacteria that we have now found in all of the Morgellons patients is called Agrobacterium. Agrobacterium is a plant bacteria that typically only infects plants. But we think that what may have happened in these patients is that they have an infection with this plant bacterium, and that then causes these fibers to grow out of these skin lesions. So in a sense, these patients are being turned into plants.
Many studies on animals have shown GMOs to be very harmful. One such study, conducted by Dr. Arpad Puzdai at the Rowett Institute in England found out that GM potato induced acute immune response in rats. It had proliferative effect on the gut cells of the rats, which could lead to cancer.
It had twofold effects. First, it started to increase a proliferative response in the gut. And that you don't like because this is possibly, I'm not saying that it is cancerous, but what it does, it can have an adjuvant effect on any chemically induced tumor. The other thing is the immune system was certainly got into high gear. And that was, we don't know whether it's good or bad, but it certainly did recognize the GM potatoes as alien.
Many other studies on animals all over the world have found GM crops to be harmful. It has made cows and pigs sterile, acute immune response in mice. Other products from Monsanto has also found to be dangerous to the health of human beings and animals. RBGH, or recombinant bovine growth hormone, was the first genetically modified product to be approved in the US. RBGH, brand name Posilac, is a hormone which increases the yield of milk from cows up to 20%, but it also causes a disease called mastitis, or the infection of the udder. Posilac, which is a product of Monsanto, was banned in Canada and Europe. Canadian scientists found out in a research that it doesn't only cause diseases in cows, but it also leaves a hormone, IGF-1, as a byproduct in the milk. IGF-1 causes cancer in human beings. The United States of America is facing these challenges from the threat of genetically modified or transgenic organisms. US being 1/4 of the Indian population, the death from cancer in the US is 1.5 times larger than that in India. Immune disorder and genetic disorders are skyrocketing there. If transgenic food crops like BT brinjal will be allowed in India, it will open up the floodgates of other such crops. The health of the people will be at even greater risk, and the younger mass, the children, will be at greater risk. It will also pose a threat to the immense biodiversity of the region. Orissa alone accounts for more than 250 native varieties of brinjal, some estimating it at more than 400. When countries like Canada, Germany, Switzerland, and the European Union are banning, labeling, or putting a moratorium on GM products and GM crops, why is India planning to launch these crops? For whose benefit? Indian agriculture is a small-scale enterprise. It is more vulnerable to the threat of takeover by large multinationals. People in India save their own seed. If contamination occurs, then the farmers will lose their centuries-old heritage of indigenous